From the University of Toronto's Leslie Dan Faculty of Pharmacy, this is the I'm Pharmacy podcast. I'm Mina Tadros. As we've been discussing, to go from a potential drug molecule to being on the shelf of a pharmacy is a long and windy process. Generally, the drug development process and life cycle goes through 5 major steps. We spent the first few episodes exploring the first 2 steps: discovery and development, and preclinical research. These 2 steps are happening deep in laboratories with no human testing. Step 3 is the clinical development phase, or the big human trials. After those, there's Step 4, regulatory review, and Step 5, post-marketing safety. We'll talk a little bit about those more in this season. But the real big jump happens at this Step 3. How do you go from the lab to the clinical development? So today, I wanted to spend some time thinking through how we pick which molecule gets a shot to win, and what that process means. It really sets up our future discussions about why and how we test in humans. It also gives us a chance to realize how cumbersome this process is. But why is it so important to pick one molecule? Well, how long do you think it takes for a new drug to get approved in the United States? So on average, from the time the molecule is discovered to when it's actually approved by the FDA, it takes 12 to 15 years. More importantly, it costs close to $1 billion to bring a drug to market. And it's also estimated that of the thousands of molecules that are looked at in the first few steps, only a few will even make it to Step 2 and then 3. And of those that make it to clinical development, only 5 to 6% will survive that phase and end up being submitted for regulatory review. So picking the right horse is important so you don't waste time or money. So how do we choose that winning horse? Because of this time and risk, many steps are taken to mitigate risk. Important pivotal work is done in those first few steps, like picking the right formulation, the correct dose, the right indication. And although lots of pivotal work can go into that, it sometimes takes a bit of luck. One story that's always intrigued me about this process is the story of sildenafil, or famously known as Viagra, the little blue pill. Now, I didn't pick this example just to make people blush, but it really does paint a wonderful picture of this process. The initial development of sildenafil was not meant for ED. It was actually meant to treat pulmonary hypertension, a deadly disease that harms many young people. So the drug works by causing vasodilation. It opens up the blood vessels. And that was the point of using it for pulmonary hypertension, to help those suffering with that disorder. But during some early clinical trial work where they were trying to figure out the right dose, they realized that some of the male patients were experiencing a strange adverse event: prolonged penile erections. But they couldn't explain why. So it was during those early trials that led Pfizer, the maker of the drug, to go back to the lab and understand this adverse event, potentially to reduce doses or change the formulation. But what they realized is that the receptor that this drug interacts with was highly presented in the erectile tissue. And this finding caused them to pivot their body of research and use the medications towards this new indication. And it would be the first ever oral treatment for ED and develop one of the largest blockbuster drugs in history. It also changed the way that we treat male health. What this story really paints well, in my humble opinion, is that to be able to pick the right horse, it takes great science, great observation, courage, and sometimes a little bit of luck. Now, if that doesn't describe drug discovery, I don't know what does. So in today's episode, we want to explore how we think through those drugs that go from lab to clinical trials. How much work goes into that process? And I want to touch on that excitement of developing a new molecule that goes from the laboratory and is finally used in a patient. To begin understanding this area of translational research, I spoke with Professor Micheline Piquette Miller, who is the Associate Dean of Research at the Leslie Dan Faculty of Pharmacy. We talked about this jump from the lab to the bedside. How did you decide which area to study? Like, what drew you to this area and the questions that are being asked? And has the questions you asked when you first started training and your graduate training to today changed?
Well, I started off as a pharmacy student. I was invited to do research in the summer in some of the research laboratories at was at University of Alberta at the time. And I really, really enjoyed doing research in the summers. And I did research in a couple of different laboratories. And I was really enjoying the research I was doing in one lab, which was pharmacokinetics laboratory. After I graduated, I decided I really liked research. As much as I enjoyed being a pharmacist, I was really intrigued by research.
You got the bug.
I got the bug. And I chose the research area was in clinical pharmacokinetics because in the research that we were doing, it was the clinical question was driving the research. And so we would identify the research question by what problems that we were having in the clinic with patients, observations that were seen in patients, and we could actually do research studies that actually delved deeper into it and determined the mechanisms behind what we were seeing in patients. And so this kind of started from at that time, kind of starting translational research. So I as a pharmacist, I enjoyed the clinical aspect of this. And as a basic scientist, I enjoyed the way I could delve into these research questions into using a number of different type of models mechanistically.
Yeah. What questions, when you first started your career as a researcher, were you asking, and how has that shifted over time?
I think that the research questions that I was looking at while I was a PhD student are still relevant. So I was looking at very specific disease states and the impact on overall drug levels. And in particular, I was looking at some drugs that were metabolized by drug metabolizing enzymes. And we were looking at changes in drug metabolism due to disease states. And we looked at, elderly, and we looked at arthritis, and we did studies in humans as well as animal models. One of the things is that during my PhD, I had a couple of questions that I thought that they couldn't be answered by changes in drug metabolizing enzymes or other types of things. And I thought actually it was transporters that might be involved in the observations, and that we'd, again, disease-imposed changes in drug levels. And I wanted to see what the contribution was of drug transporters to these changes. I started looking at some basic models of diseases such as bacterial infection, viral infection, and I looked at a number of different types of tissues. This led me sort of to focus more on the placenta. I had been working with some other researchers doing some research in ovarian cancer, and I was very much interested in women's health and had noticed that there was an absence of research and studies in pregnant women. And in particular, there's a number of changes that women, while they're pregnant, undergo, and the pharmacokinetics of drugs are really not that well understood in pregnant women. Also, the impact of these changes on fetal exposures to drugs is really not well understood. And so my focus sort of changed into looking at overall impact of maternal conditions on maternal and fetal levels of drugs and on maternal and fetal health.
So it's like your work is really on the cusp of like clinical impact. And it's how has it been that sort of, we talk about translational work, but you're really doing it. And how's that transition been from the lab to the hallways of the hospital or the clinic or whatever? Like, tell me about that dynamic and was that always your intention from the very beginning of your work?
Well, I do have a number of clinical collaborators at the hospital. So I've got a couple of the OB-GYN collaborators who help me recruit patients. And we do have discussions in terms of what we're finding, what types of diseases. And we have discussions in terms of, like, let's see, what does that mean to drug therapy in these pregnant women? Is there particular drugs that you would recommend or avoid in these patients? So, and we have, we've also run a few systematic reviews looking at whether or not there's any signals of information from clinical studies that have been done in pregnant women to see whether, certain drugs have actually, raised suspicion in terms of that there's been poor outcomes with either the mother or child. So we'll use a number of different types of clinical approaches to see if we can answer this question.
Do you find that is it hard to land on the same question if someone's coming in from the clinical side and you're coming in from maybe a translational science side, or are you speaking the same language from day one? And it's like when you find collaborators working in this space?
Well, I think being a pharmacist and having that basic ability to translate into different topics, like I worked as a pharmacist for several years. I'm used to talking to physicians, and throughout my PhD and my postdoc and in my career, I've had physician, clinical collaborators. And so I haven't found it difficult. I find that if there seems to be some sort of knowledge gap or misunderstanding, I know as a pharmacist, I've been trained to step back and rephrase. Yeah. Because we're used to trying to explain sometimes some difficult concepts to patients, or and sometimes if I'm coming at it at too much of a scientific angle, and it's not really, they're not really catching it, then I know that I need to step back and come up with a different terminology and a different way of phrasing it, and maybe analogies.
Given the breadth of your work, like animal models all the way to clinical work, not that you have to pick a favorite, but which part of your work gets you the most excited these days? Like what's the most exciting part of?
I think it's when we find that, so sometimes we actually start our studies with looking at animal models first, and then seeing whether or not we have justification to move into humans. So, this is the part of being translational research. You need justification to do human studies. And I think one of the most exciting things is when we've done the animal studies, we've come up with the justification to do some of the human studies and get the human samples. And then we start analyzing human samples, and we find the exact same things that we found in the samples from our animal model. And it's just like, wow, that's amazing.
Yeah, your hypothesis was proven true.
Yeah, it was proven true. And you're just like, okay, so we know we're on the right track. Let's keep on going. And I
feel like those eureka moments are why people do science.
Yeah, exactly. and usually in those cases, we've actually thought of, mitigation strategies that we've seen certain changes, and we've kind of come up with, we understand the cell signaling pathways. So we know what has been altered because of this condition or disease. And then we've come up with what could we use to prevent these changes? What could we feasibly administer to a pregnant woman, and that would be, would not be harmful. It would basically be benign. And can we prevent the changes that we saw?
To continue our exploration in the area of translational research, I spoke to a researcher who's had some recent success in the space, Professor Raymond Reilly. Dr. Reilly works in the field of radiopharmaceuticals and uses them to treat cancer. Most recently, he partnered in a groundbreaking human trial that was able to successfully deliver a cancer drug across the blood-brain barrier. Here's Dr. Reilly.
I work on radiopharmaceutical science. So radiopharmaceuticals are drugs that are mostly used for diagnostic imaging of various diseases, but they also have great potential for treatment of diseases, especially cancer. And so in my research, I am developing radiopharmaceuticals for both diagnostic imaging of cancer as well as treatment of cancer.
So currently, nuclear medicine is most used for what? Like if a patient is getting treatment now outside of research in a standard of practice, what are the most common uses for nuclear medicine in the current state? So
nuclear medicine is actually mostly a diagnostic imaging area, but it has, as I mentioned, great potential for therapeutics. So most patients going to nuclear medicine are having an imaging study done, And those imaging studies are for a wide variety of diseases, all the way from heart disease to infections to cancer to neurological diseases, almost anything. It's a very wide to pulmonary embolus, for example, which is a blood clot in the lungs, all kinds of different options for studies in nuclear medicine.
So walk us through an example of is this an area that you're thinking about currently in your work? And what would be an example of how it can be a diagnostic and a treatment at the same time?
Yeah, so actually, if it's all right, I could actually tell you about the story surrounding the radiopharmaceutical development for the clinical trial at Sunnybrook, which really
I love stories.
Exemplifies this idea of a theranostic.
Amazing, yeah. Walk us through it.
Okay, so the radiopharmaceutical that we developed for the clinical trial at Sunnybrook Health Science Center is actually based on a drug called trastuzumab, or Herceptin, which is a drug used to treat breast cancer. A particular type of breast cancer called HER2-positive breast cancer. And so I was very aware that this drug was used to treat this type of breast cancer, but I was also aware that not all patients respond to treatment with this drug, and also that some patients who initially respond to treatment quickly develop resistance within a year. So I thought, how can we improve the efficacy of trastuzumab and overcome the resistance mechanism? So being in the area of radiopharmaceutical science, I thought perhaps we could attach a radioisotope to trastuzumab that would allow for its greater effectiveness for treating HER2-positive breast cancer. And so initially I developed a radiopharmaceutical based on trastuzumab that involved linking trastuzumab to a radioisotope called indium-111. And this is a radioisotope that's been used for many years in nuclear medicine for imaging purposes. But it also emits very low energy and short range electrons called Auger electrons. Most researchers in the field didn't pay much attention to those electrons, but several years ago I started to look into the potential of using Auger electrons for treating cancer. So I decided in this particular case to link trastuzumab to indium-111 in order to use the Auger electrons emitted by this radioisotope to treat breast cancer that was HER2 positive and improve the response compared to trastuzumab. So the radiopharmaceutical was developed by a previous graduate student who graduated from my research group a few years ago. And he found actually that, in fact, the radiopharmaceutical, this form of trastuzumab linked to the radioisotope, was far more effective at treating those tumors and in the mice, and also could overcome resistance. So tumors that were resistant to trastuzumab still responded to the radiopharmaceutical treatment. Based on those promising results, we thought, could we formulate a clinical formulation of the radiopharmaceutical that would allow us to do first-in-human studies to test its potential effectiveness for treating HER2-positive breast cancer? So I actually leveraged the experience and expertise that I had gained as a nuclear pharmacist for 17 years to formulate a clinical form of this radiopharmaceutical for first-in-human testing for treatment of HER2-positive breast cancer. Now, all stories in research there's never a straight path from point A to point B. There are many circuitous paths along the way. And so we had developed the radiopharmaceutical, the clinical radiopharmaceutical, and also tested its safety preclinically in anticipation of doing a clinical trial of the radiopharmaceutical for treating HER2-positive breast cancer. As it turns out, the clinical trial did not proceed, but fortuitously, at the same time, I was contacted by a team at Sunnybrook Health Science Center who wanted to have a radiopharmaceutical form of trastuzumab for imaging purposes, not for treatment, for imaging purposes, to study the delivery of trastuzumab to brain metastases in patients with HER2-positive breast cancer, because the team at Sunnybrook was studying a revolutionary new technique called MRI-guided focused ultrasound, which had the potential to disrupt the blood-brain barrier and allow trastuzumab to enter the brain to treat tumors, metastatic tumors from HER2-positive breast cancer that had formed in the brain. The team at Sunnybrook had already determined that they could, in fact, disrupt the blood-brain barrier by using MRI, but what they really needed to know was whether or not they could deliver trastuzumab more efficiently to those tumors in the brain after MRI-guided focused ultrasound. So I decided to repurpose my drug, which is another concept, I think, in pharmaceutical science, which is repurposing of drugs for other applications. And so knowing that indium-111 had been used for years in nuclear medicine for imaging, I decided to repurpose the clinical radiopharmaceutical as an imaging agent for the study at Sunnybrook to evaluate drug delivery to tumors in the brain. But based on that collaboration, I became more interested in developing radiopharmaceuticals for treating brain metastases in patients with HER2-positive breast cancer. And so I submitted a grant application to the Canadian Institute of Health Research, along with the team at Sunnybrook, to develop another form of trastuzumab, which is trastuzumab linked to a radioisotope called actinium-225. It's a very powerful radioisotope, and study that uptake of that radiopharmaceutical preclinically in brain metastasis in a mouse model, with the idea that we could combine in the future MRI-guided focused ultrasound with this new form of the radiopharmaceutical to improve the treatment of HER2-positive tumors in the brain.
An amazing story. I have a ton of questions. I think that the first thing I think maybe that our listeners may not know, especially if they're kind of new to the field, is why this is so incredible. Is that maybe you could tell us a little bit more about the blood-brain barrier itself and why why is that so exciting compared to perhaps other uptakes in the body of being able to cross it?
Okay, so this gets back to the idea of trastuzumab as a treatment for HER2-positive breast cancer.
Yeah.
So the drug was developed and approved in the late 1990s, and it's been used for 2 decades now for treatment of HER2-positive breast cancer. It's one of the most successful drugs ever developed for this type of breast cancer, except that it's actually, because of its success, patients are living longer. And what used to be a very rare occurrence of brain metastasis from HER2-positive breast cancer is becoming more frequent now because patients are surviving much longer, even with metastatic breast cancer, because of the development of trastuzumab and other targeted therapies for HER2-positive breast cancer. So now the patients are presenting with tumors in the brain, and it's very difficult for trastuzumab to treat those tumors because it's a large molecule drug, it's a monoclonal antibody, and those types of drugs don't penetrate the blood-brain barrier very readily. As a result, the concentration of the drug in the brain, for example, is approximately 1,000 times lower than it is in the plasma in a patient. So there's a major difficulty in delivering trastuzumab across the blood-brain barrier in order to treat those tumors in the brain. And unfortunately, because of that, patients ultimately succumb to brain metastasis from HER2-positive breast cancer. So the team at Sunnybrook has been working for actually more than a decade on trying to improve drug delivery to the brain by using focused ultrasound. So this is ultrasound energy that is applied to a region of the brain, and it temporarily disrupts the blood-brain barrier and creates an opportunity for an intravenously administered drug such as trastuzumab to penetrate the brain and reach a tumor in the brain for treatment. And so this is really a major breakthrough in the treatment of HER2-positive breast cancer, especially for metastasis in the brain. And it really was my privilege to collaborate with the team at Sunnybrook to use the radiopharmaceutical that we developed here at the Leslie Dan Faculty of Pharmacy in order to prove that approach had a lot of promise for treating tumors in the brain.
No, it sounds incredible. When we learned obviously about the blood-brain barrier, it feels like it's like an iron wall, right? Like most, you kind of assume that many things can't cross. So, it does sound like it opens up so many more opportunities to be able to do that, especially when trying to treat like what you described as a sort of a terminal and very serious affliction that happens to some patients. One thing in your story that I want to dive a little bit deeper into is you discussed the great work that was done by your PhD student on the mouse models, and then first in human testing. That feels like a pretty big leap. Like, what does it take to jump from the really great results in a mouse to getting the approval to test it in a human? Can you tell us a little bit about that? And like, what kind of timelines is that? Is that weeks, months, years? Like, walk us through that kind of gap between those two steps.
Okay, so the preclinical studies that the PhD student worked on took about 4 years to demonstrate the effectiveness and safety of the radiopharmaceutical for treating HER2-positive breast cancer. But you're absolutely right. In order to take a radiopharmaceutical that you make on the bench in very tiny quantities for a mouse and scale that up to something that is could be used in a clinical trial in a patient, and also that is safe to use in a clinical trial in a patient, and that could receive Health Canada approval for proceeding to a clinical trial in a patient is a major challenge. I feel like the time that I spent working as a nuclear pharmacist at UHN prepared me for that challenge.
We've spent a lot of time discussing the effort and complexity behind the discovery and development of drugs. This research can take years and even decades before it's put to test in humans in a clinical trial. Similar to drug discovery, the process of developing diagnostic tools follows a similar path, but often follows very different timelines. Meet Dr. Jennifer Doucet, a postdoctoral fellow in Professor Keith Pardee's lab at the Leslie Dan Faculty of Pharmacy. Dr. Doucet and the Pardee lab specialize in the development of disease diagnostics, working to navigate a different set of hurdles to get their research into the hands of patients. Here's Dr. Doucet.
Broadly speaking, a lot of the work in the Pardee lab is focused on a couple of different themes within synthetic biology. So the theme kind of that my research is mostly focused on is using cell-free systems for diagnostics. Very relevant with like the ongoing pandemic. And actually when I joined to work on a grant under the COVID-19 pandemic response program. So that was run by the federal government to come up with new diagnostics. And the way that our diagnostics work is that we're focused on kind of trying to kind of decentralize it. So right now with molecular diagnostics, most of the tests run are like qPCR based. So they're run in centralized reference laboratories. It might take some time, especially early in the pandemic. There was some supply issues with some of the reagents and even some of the staff to run it. So the systems that we're kind of working on are essentially use these mechanisms called toehold switches and cell-free systems. So toehold switches are essentially a mechanism to detect your pathogen DNA or RNA, which in response activates the expression of a reporter molecule. And usually these reporter molecules are fluorescence proteins or some sort of colorimetric output. The project that I'm actually focused on is looking at an output that's glucogenic. So the reporter is actually an enzyme that converts a precursor to glucose, which could then be read on an off-the-shelf glucose meter.
Oh, that's cool.
Yeah.
So is the idea there that you could just repurpose a glucose meter? Like you could just use the same strips?
Yeah, you can use the same. So the way that it's run, we use a $10 glucose meter from Bayer. Yeah. And we run it on the on the strips. And it works quite well. The test takes probably about 2 hours. Hands-on and hands-off time. And actually, that work was published earlier this year, and it was kind of spearheaded by a graduate student in the lab, Evan Amalfitano. But what the current project that I'm on is focused on is actually taking that assay and trying to automate it. So this is in collaboration with the NRC and the microfluidics facility in Boucherville, Quebec. So we've had the pleasure of kind of working with the team at the NRC to try and kind of solve this sample prep and automation issue from both kind of the engineering side and the assay side.
So what is the issue? Walk me through this. So what is the sample issue? Is it the size of the sample you need, or? So
the issue is that it's like technically challenging to run the test. So it's basically what you do is you have to extract the RNA from the virus. So you have to find a way to kind of crack open the virus and purify that RNA. Usually what we use in the lab are what are called spin column based extraction prep kits, where essentially like you're taking multiple steps where you're pipetting small amounts of liquid from one tube to another. You're putting it in a centrifuge, and it just doesn't really translate well to
And that's why it was taking 2 hours before.
Yeah, and then there's also time for the assay to run.
Right.
And then again, even just for running the assay, again, you're taking small amounts of volumes of liquids, you're mixing them together, you're putting them in a machine, and it's all very like hands-on and very manual. So the idea was, well, let's de-skill this assay so that anyone could run it. So like you could go to a border crossing and the border guard could collect your saliva, and then the test would just run automatically. And you'd be able to find out like whether, this person might have COVID or not, or any other infectious disease.
So going from the original, like kind of like 2 hour process to now, how much of a change has it been? Like, with this prototype, what are you envisioning the process would look like now?
Yeah, so the process is still going to take about 2 hours because it's a biological system, like the DNA amplification process, as well as, the running the assay through the cell-free system. Like it does take time because, the way the cell-free system works is you're basically translating, you're going through protein translation.
Right.
So it's going to take some time just for the mechanisms to work. But it's like going to be the sort of thing where you can walk away. Like you add the sample and you can walk away. So it's a sample to answer system.
That's awesome. So like what you've sort of eliminated is that whole other process that somebody who had to be skilled to do it.
Yeah, exactly. So normally like you would probably need someone, in a lab or with a lab facility to run it. Yeah. And it was still going to be, maybe cheaper and like easier to run than say like QRT-PCR, but it's, I think that sample, like that sample to answer kind of system where you're doing sample prep and running the assay in an automated way is definitely a key part of it.
So is the sensitivity and specificity of this compared to the PCR? Is that sort of the gold standard of what you would compare to? Yeah,
the goal is to get it to a similar level of sensitivity to QRT-PCR.
Yeah.
And then a similar level of like reliability, because I think some of the problems that we've seen with uptake of, rapid tests or even like some of the lateral flow tests is that like they're not perceived to be quite as like reliable and they're not quite as sensitive. So our goal is to really like have this system, match or at least be very close to qPCR in terms of sensitivity. And we have plans for our validation assay to actually, run our tests side by side with qPCR to do that kind of direct one-to-one comparison.
So I want to go back to developing prototypes. So that's where you guys are at. You're developing a prototype. Walk us through what would happen next. Like, how would you get this into border control pharmacies around the world? Like, what would be the next steps that are needed? Because I'm sure that you don't have enough prototypes to make a dent in the problem we're dealing with.
Yeah, so there's definitely like a long way to go. I think like for me, having had a little bit of experience working in industry and seeing like, so I was very fortunate to be exposed to the, 510K pre-approval process and the 510K approval process and being involved in clinical evaluation of diagnostics technologies. Right. And it is a big undertaking.
And that's to get Health Canada approval for something, or the FDA approval?
Yeah, FDA approval. So 510K is FDA. Health Canada, I'm a little bit less familiar with their process, but they have a, I'm sure, similarly robust process. And so there's a lot that needs to happen in terms of, the kind of validation that we can do in an academic setting, is definitely like different from the kind of validation that you can do in on a larger scale. So the next step would really be kind of taking things to the next level. So doing more mass manufacturing, more figuring out manufacturing processes, and then planning validation studies, planning clinical trials. And this sort of thing would also all be planned and kind of in collaboration almost with the regulators. You have this pre-submission process and this active review process. And I know that's changed a lot during the pandemic because a lot of things are getting emergency approvals. But you still have to do quite robust clinical evaluation and assessment of, the performance of your assay. You want to make sure that it's running. There's a big difference between running an assay 3 times for a paper and running an assay 3,000 times.
Yeah, well, that sounds like scalability. So would once you've, as an academic now in this once you have a prototype you believe in, do you find partners? Is that often what happens, or do they create companies? What are sort of the logistics of doing something like that to be able to scale to meet these requirements, which sound pretty robust?
Yeah, so there's, I guess there's a couple of different ways to do it. For us, we've really worked closely with people at our Innovation and Partnerships office here on campus. So they would kind of try and bring some of this IP that we've developed and kind of bring it to the attention of potential companies that might be interested. We already have at least one Canadian company that's actually very interested, who sat through several meetings with. There's also the option of, commercialization through the creation of startup companies. That's another,
it's an exciting space to be in. Yeah,
it is. It definitely is. So all this to say that there's probably more than one way to crack an egg.
Yeah.
And kind of look at different ways that you could, potentially take this out of the academic lab, because at a certain point you do need to kind of go that next step and take it a little bit further in a way.
Is that intimidating, or does your past experience working in the private sector kind of help you? Is it intimidating going from a lab to sort of the business world in a way?
Yeah. I think that it's definitely a different skill set. Even I think, my industry experience definitely helps because being familiar with what the requirements are going to be and what the right way to do things, how to work in kind of under GXP processes. So that's, good laboratory practices or good manufacturing practices, and kind of being familiar with that, helps what you're getting into. But I think that the business side and pitching an idea, coming up with some sort of, market segmentation, value proposition, those are all things that I've had to learn, I think. And, I think UTIST has been great for that. And, even just there's a lot of resources available just for anyone who's interested in starting something up. And there's definitely a lot of students, even in this department and on campus, who are coming up with great ideas and trying to validate their ideas in a business setting.
Yeah, I know. I can imagine just being in the pharmacy building, looking out the window and seeing Mars there. It almost feels like it's, in a way, it's just the connection kind of feels like the perfect location to be able to go from that. I don't know how you guys do it. I think it would be intimidating to kind of switch that gear from research to business.
Yeah, it's definitely a different experience, and we're definitely in the very early days yet. But you do your best to try and that's one of the things one of the reasons why I wanted to join, I guess, Keith's lab is that he's not just focused on doing some really unique and new research and getting a really nice paper. He genuinely wants all of these technologies to kind of succeed and to try and to actually leave the lab and actually go out into the real world and help people.
The jump from bench to bedside is really a balancing act of risk and benefit. This balancing act is built on a strong foundation of many years of basic science. And to make this jump, this leap, is huge and takes years and years of evidence building to ensure that the risks and benefits are well balanced. As we saw through all three examples, it's not taken lightly and requires collaboration with many different types of scientists, clinicians, and navigating a series of important regulatory frameworks. We saw that depending on the area of work and the risks and benefits, that it could take different types of timelines, in many cases lasting years, and in some cases taking months. This essential step is massive and is the start of the next phase in the life cycle of the drug. It's the leap into clinical research. This episode of the I'm Pharmacy podcast was produced by Steve Southon, Kate Richards, and me, Mina Tadros. It was edited by Steve Southon with musical support from Steve Southon and Diego Martinez. Special thanks to Professor Micheline Piquette Miller, Professor Raymond Reilly, and Dr. Jennifer Doucet. We'll be dropping new episodes every month, so be sure to subscribe to wherever you listen to podcasts. Keep asking questions, stay safe, and catch you next episode.